welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
I'm Sylvia Earle. I'm a National Geographic explorer, founder of Mission Blue, and I'm an ocean elder. Thank you, ocean elders, for sponsoring this program.
Yes, and this is our show, Dive In, where we host informal and open conversations with the ocean community on topics of wonder and interest. We're glad to be back and hope everyone has made some time to get outdoors. You can write or ask us questions by putting them in the Q&A box, and later on in the conversation, we'll get through as many as possible. And there'll also be some links in the chat for further information. And before we get underway, let's remind everyone that the world
is
blue. It's very blue. And today we're very excited to have Doctors Alyssa-Lois Gehman and Melanie Prentice talking with us about their work at the Hakai Institute in British Columbia, Canada. Alyssa is a marine disease ecologist, adjunct professor at UBC's Institute of Ocean and Fisheries, and a member of the Biodiversity Research Center. She's particularly interested in the interactions between infected hosts and their communities, as well as the impact of temperature on host and parasite interactions. And Melanie uses genetic and genomic tools to answer complex ecological questions to meet applied conservation and wildlife management goals. And most recently, she's been focused on controlled challenge experiments and genomic datasets to improve our understanding of wildlife epidemiology. Welcome, both of you.
Yeah.
Thank you.
Well, while I work on sharing the slides, tell us how each of you got interested in finding a way to work with echinoderms.
Okay. Do you want me to start?
Yeah, go for it.
So my first memory of a sea star was, I think, from 8th grade. And it was from an 8th grade kayaking trip in the Desolation Sound in British Columbia. And I just remember seeing probably ochre stars, so pies aster, because I remember purples and oranges. I'm sure I would have also seen sunflower stars, so because they would have been around. And I just remember that the water was clear enough, and they were just all over, and it was really kind of remarkable how much color there was there, and that was pretty much what I remember from. I was in 8th grade, and then I had an incredible high school marine science class. So I was really lucky and got to learn about invertebrates in high school, and got really excited about the ocean. I learned how to scuba dive, and yeah, and then I think that was pretty much the point at which I was like, I'm going to do marine science. I will study invertebrates, and I studied invertebrates in general in the ocean, but I have a pretty big emphasis on sea stars at this point in my career. And maybe
Melanie?
Yeah, I mean, quite a different story. I grew up in Toronto, in Canada, very distant from the ocean, very distant in some ways from nature. And I wasn't really connected to that until I moved north, where I ended up doing my PhD. And my background from my undergraduate degree was in forensic science. And that's where I learned a lot about genetics and how powerful genetics and genomic tools can be to understand the natural world around us. And so I very quickly shifted into a more of a conservation-based angle for my doctoral degree. But it wasn't actually until I started postdocing after I got my degree where I started working on marine systems. I moved to the UK, and I was working in the intertidal there and on this big project looking at development in the intertidal and how that impacts intertidal species. And that was like the first time I'd lived by the ocean. It wasn't just a vacation, like it was really my everyday life, and being able to just kind of wake up in the morning and hear it and smell the sea and be right there. And that was like totally life-changing for me in a way that I'm sure my parents hate, because it meant I was never going to go back to Toronto. But yeah, and then I kind of came back to North America during the pandemic, and I was kind of putting the pieces together. What do I want to do? How do I make this into a career? And I'd worked on some disease systems, and I'd worked on some marine projects, and I kind of put those two things together as something that I thought I could really hone in on and focus in on for the rest of my career. And that's when I actually reached out to Alyssa and they were happy with this project. Yeah.
Well, the sea stars are such iconic animals, of course, to divers, tide poolers, snorkelers, beachcombers, and that gorgeous symmetry that they have. It's just it made it just such a shock, I think, to the system of so many people when they were suddenly dying en masse.
Yeah.
A real puzzle about why. And it took your kind of inquisitiveness and skill to unravel the questions.
Because it first started around 2013 when they started to be noticed largely in big areas. Is that about the right time frame?
Yep. Yeah, it started around 2013, actually in the sort of BC, Washington area. And then very quickly, it was documented down in California and then sort of slowly filled in the coastline. Maybe not that slowly, in the coastline until, we all the way from Mexico to Alaska.
Wow, unbelievable. And yeah, it was just all the people that come in through our dive center and so forth, they're just practically in tears, thinking about the loss of these animals and particularly the beautiful sun star. Just such a huge iconic animal. I mean, they're huge.
Is that like the stuffed one you have behind you?
Yeah.
Yeah, this is a sunflower sea star. It's the species we primarily work on right now, and also the one that's like most heavily impacted by wasting disease. So billions of these guys were lost to this disease, and this is like actually to scale of what these species look like. They are truly massive and gorgeous.
And this is sort of an average sized adult. Like they get bigger than this.
Yeah, they're just enormous.
Yeah. Not something you'd think of if someone says like describe a sea star or draw a sea star. Probably not what you're going to come up with, but is a sea star nonetheless.
I mean, usually think of 5 arms. So it's a pisaster, and then it has 3.14.
I like that.
What about disaster?
But these big beauties, what do they normally eat? And if they disappear, do the things they normally eat increase?
Yeah, so the thing, there's sort of like 2 answers to that question. One of the things is they eat almost everything that's on the floor of the ocean. So one of the things that I remember the most about learning about sunflower stars is almost every benthic, so anything that lives on the bottom, has an escape response from a pycno coming towards them. And so when I was taking my inverts class at Friday Harbor Labs, we would sometimes stick a little pycno arm in a tank and then everyone would explode.
Hey.
Like there's like scallops. It was the one that was the most fun was a tank with scallops in it. And you stick the arm in and then they start swimming around. And if somebody's leaning too close, they'll get wet. And so they're incredible. They eat so many things. But what we found out after wasting was some of the things that they eat actually matter quite a bit. Like they don't, yeah. And so maybe you want to talk a little bit about the urchins.
Yeah. One of the species that they seem to eat a lot of are sea urchins, which for those unfamiliar, they're like tennis ball sized, spikes all over them. Kind of funny to think of a soft-bodied sea star actually being able to ingest something like that, but they do eat quite a number of urchins. And I think that's something that we kind of learned secondhand post disease outbreak is like how important this species is to controlling urchin populations. Because when billions of sunflower stars died from wasting disease, that really removed the pressure of predation on urchin populations, which absolutely exploded in number. And then you have all of these urchins, which are grazers, eating things like kelps, and essentially removing like swaths of kelp forests. So places like Northern California, you see losses exceeding 95% of the kelp forest there. And the loss of these predators, among other things, like we know sea otters are also important in this scenario, but sunflower sea stars seem to play a really big role in kind of keeping the ecosystem in check so that everybody's kind of in balance, basically.
Yeah,
But what's curious is that when there was a sea urchin die-off in the Caribbean not long ago, the as you say, they tend to graze on algae. But when the sea urchins disappeared, the algae started to overtake the reefs.
Mhm.
Yeah.
Everybody has a place
Yeah,
with
it's a balance.
a coral reef, and if you take out all the fish, or even some of the fish, or a starfish, or an urchin, you're you're dismembering something that is closely connected as a system. It's like removing chunks of your computer.
that's right.
Yeah.
Who needs this little wire?
Yeah. You don't really need the return key, you know. Yeah. The cap lock for you. Yeah.
I
The
think that's something that's so like impactful about sea stars as well is there's this, concept in ecology, keystone species. Many people might be familiar with it. And you might think about terrestrial species like wolves regulating populations of deer in Yellowstone National Park. And that whole concept of a species being like disproportionately important to its habitat was actually created around research on sea stars in Washington State. So, like, they are the original keystone species.
predator.
They're just so important to the ecosystems that they inhabit and keeping all these other species kind of in check in the right ways.
And is the map that we're seeing here, is this where is this showing percentages of where the sun stars particularly died off?
These are declines, yeah. So that's how much we've lost in those areas since the
Wow. So 100% along the California coast.
Yeah.
yeah, functionally pretty much gone.
And down very, very far south as well. Um, and and the temperature kind of played a role in this as well. Some of the warm water blobs, as they were called.
Yeah. So there's a lot of correlation essentially at this point between warm temperatures and the disease outbreaks. And that's actually, those are, we're getting to run those experiments right now because before we knew what was causing wasting disease, it wasn't really possible to ask, like, what is this relationship? But now that we have a causative agent, we're actually we are running and going to continue running experiments where we're sort of crossing disease with the temperature and trying to understand, our hypothesis is that their warmer temperatures exacerbate the disease and potentially make the pathogen grow faster. And so maybe that's what's happening. And that's why we're seeing sort of these bigger disease outbreaks at a much wider area.
Cause of the disease, but it just makes it worse.
Yeah, exactly. And there is, so that picture, if you go back just one, this picture is actually taken probably not that many years ago. So this picture was taken after the wasting outbreak in these refuge populations that we found in British Columbia. And you can see, like, there's a diver behind that sea star. So that's a full size adult sea star.
Yeah.
And these are in the fjords in British Columbia. And we did some work. We've been doing some observational work in the field, both in the fjords and then in sort of islands that are near the fjords that are more in the open ocean. And it's just incredibly different looking. When you're in the fjords, you see these massive pycnos. They're actually at pretty high densities. Like, it looks like the before times. It's pretty incredible. And then you go out into the outer islands, which are just a couple kilometers away. And they're just little tiny ones. And like places they used to be, they haven't come back. And so we compared what was different between those two locations, and it was temperature. So those ones that are in the fjords, the areas that they live in are cooler and cooler by several degrees than the ones in the outer islands. And we see disease in both locations. So the disease, it's not that there's no disease in the fjords, but we don't see the population loss after we see the disease. And so I think there's just not as much mortality.
Yeah. Yeah.
So how long does it take for a little planktonic one of these creatures to get as big as the one here? And how long do they live? Do you know? I mean, normally.
Yeah, this was when so I was part of the group that sort of got together to try and put together the data we needed to get them listed with the IUCN. And that how long do they live is one of the questions we're required to answer. And quickly became clear that like we don't know particularly well. Sea stars they're amazing. They can grow, obviously. They can spend a year just not growing and then grow the next year. So there's no like real, I mean, there might be a pattern, but we don't know what it is yet. And they can shrink if they want.
Oh, wow.
And so it makes it, we've made some estimates and our guesses based off of, I don't know, there's some that have lived 30, 40 years in aquariums. And based off of growth rates, we feel like sea star that's about that size, maybe between 20 and 70 years. Yeah, like we're not, I'm not super accurate, but it's a long time.
Yeah. Some sea urchins are known to live for a century.
Yeah.
So it's in
the other big problem is that it's really hard to tag sea stars.
Yeah, I was just wondering about how would you actually go about that? Because they're pretty good at sort of cleaning themselves and keeping themselves free of debris, and they'd probably be super annoyed with a tag.
maybe
You know?
they can eDNA them.
Yeah. So, I mean, I think people have tried to tag arms. They just drop their arm.
Yeah, they're just like, be gone. Grow a new one.
But one thing, there is a researcher at Friday Harbor Labs who is looking into taking pictures of the surface of the sea stars and looking at the spike patterns relative to where the madreporite is. And so I think they can get individual from that now. And so the thought is that potentially we might be able to photo ID individuals.
And so use some computer assistance if you get a lot of them.
Yeah, definitely. You get a little
like the
photo recognition.
Use NASA's star crunching technology to identify and distinguish whale shark spots. I mean, there are programs that could really be helpful for distinguishing individual stars.
Yeah.
Do you say starfish or sea star?
I say sea star, but I don't have a problem with either.
It seems to be the typical thing. Some people are like, oh, it's not a fish, but it's all right.
Although I have noticed recently that if I say sea star, people might not know what I'm talking about. And so I've like, maybe I should go back to saying starfish just so that I don't have to define myself.
Yeah.
Just try saying sea lily though,
Way too far. Way too far.
Basket star.
Basket star, yeah.
Basket stars are so amazing.
They really are.
We do say pycno quite a bit, which to clarify by that, Pycnopodia helianthoides is the Latin name for sunflower sea star. So it's all pycno, sunflower stars. It's all the same.
Yeah. So this kind of shows the course of degradation, the different stages of the disease.
Yeah. There can be like a lot of different disease signs. So there's like potentially up to 26 species along the Pacific coast that are impacted by wasting disease. And disease signs can be super variable depending on what species you're studying. But for sunflower sea stars, because we've studied them in the lab the most intensively, the two disease stages that we see like characteristically in a trajectory as we would expect are arm twisting. So that's the middle photo there. They kind of like twist their arms up in almost like a corkscrew fashion. And then armatotomy, which basically means their arms will drop off the central disc. And you can actually watch them walk away. It's like pretty eerie.
It's spooky. Yeah.
It is really spooky. And they can stay motile, like the arms can stay attached to surfaces or walking around for like, I mean, we haven't tested how long, but like a day, 2 days, easy.
Yeah. And when you're diving, and maybe you guys have seen this, but you will sometimes just see arms around. It's something we sort of anecdotally have shown that the arms can be contagious on their own. And I think about this in the context of being the parasite.
Yeah.
Like, especially when you're thinking about these sunflower stars that can have up to 24 arms. and their arms can walk several meters away from their body, and it's contagious at that stage. It's like a pretty terrifying disease situation.
Yeah, 100%. And, you used to hear the stories about fishermen would get mad at sea stars, and they'd
Eating
like
mussels and things.
eating mussels and things. So they'd like cut them in half and throw them, the two halves, back in the ocean. But as long as the central disc is quasi intact, then they can regrow. Is that accurate?
Yeah, so these ones, I'm not sure how much of the central disc you can cut before so their arms certainly can come off and they'll regrow them. I'm not sunflower stars may not be able to regenerate if you get too much of their central disc.
Yeah.
But they can and do regrow their arms quite a bit.
All right, so we're doing this controlled challenge experiment.
Yes. Yeah, we're getting into sort of what we did. Basically, when we started when we got into this project, the question is, why are they dying? That was pretty much where we were, and so this is the set of experiments that we started with, and also led us to the final to the answer. Do you want to describe where?
Yeah, sure. I can kind of take you through it a little bit. So I guess before you actually know what's causing your disease, we're reliant on having some sort of pathogen present in our experiments. And we obviously couldn't go out and culture a bacteria or anything because we had no idea what we were looking for at that point. And so that meant we were using kind of tissues or fluids from wasting sea stars that we saw in the field as a kind of source of exposure for our experiments. And so in this experiment, we took coalomic fluid, which is like basically a blood of a sea star. It's essentially seawater with some immune cells circulating in there. And we created 2 treatment groups. So we exposed half of our stars to just this raw, untreated coalomic fluid taken from a wasting sea star. And then the other half we kind of boiled, and that kind of presumably kills off or inactivates any pathogens or toxins that are present in that fluid. And that gave us this kind of source that we could use to expose our sea stars without actually knowing what pathogen we were looking for at this point. And then to start our experiments, we sample all of our individuals, and that gives us a baseline of what is in these sea stars before we actually introduce anything new. So like, what is there that we can kind of use to exclude some culprits from our list? And so everybody gets sampled, and then we perform our inoculations where we're exposing them to either the disease or our control group, not the disease. And then we kind of sample throughout the course of that experiment. So when they're showing disease signs, we take samples, assuming there might be some pathogen there. And we, of course, take samples from our control stars as well as a sort of another way to kind of eliminate what's in our healthy stars, what's in our sick stars. And that's where we're hoping to kind of come up with a list of different agents that we might suspect are causing this disease. And so that was the very kind of basic general overview of the experiments that we used to identify the pathogen. And we did that then with genetic data sets, which I think we show some of that data in the next few slides.
Carry on here.
Yeah, and I should say that this experiment was so great, and a lot of our experiments turned out to be super helpful because we had such clear experimental results. So when we had 100% mortality in the stars we exposed and expected to die, and 0% mortality in our controls, and that gave us like some really rich data sets where we had really conclusive results that we could use to dig in to try and find a pathogen.
Wow, 13 days. That's rough.
Really fast.
Yeah.
This is a presentation that I give, and whenever I'm giving it, I make sure Melanie did the genetics part of this data set. So Melanie is on this slide.
Gets the credit, yeah.
She gets the star.
Yeah. You're holding one up,
Yeah. So the way like the utility of genetics and genomics, I guess, in these types of investigations is being able to characterize like what's in these samples when you're dealing with things like viruses and bacteria that you can't see with the naked eye. So what we can do is we can take samples from all of these stars and sequence everything that's in them, and then use all of that data to try and find out who's there, what's there, what's in what sample. And so that's what we did first. We started with a data set called metatranscriptomics, and I will not go into that aside to say that this is like kind of painting with like a broad brush. So you should be able to get viruses and bacteria and protists and all the potential major pathogen groups. And so we created this plot here, which is any 2 points that are close in space have similar microbial communities. And basically when you color them by their metadata, on the left in orange, those are all samples that were taken from healthy stars. On the right in blue are all samples taken from wasting stars. And you can see they form like two very clear clusters. So there's something in the wasting stars that's not really in the healthy stars. So we wanted to know what that is. And when I put those little arrows on there, that was the first time we'd ever seen the name Vibrio pectinicida. And when I went back and looked at all of these like hundreds of thousands of sequences that we had, all like Vibrio pectinicida was showing up in every single one of our wasting sea star samples and none of our healthy sea star samples. So that was the first like, whoa, we might have something here.
Eureka.
Yeah, exactly. Yeah, and then the other type of sequencing data set, maybe I'll just kind of breeze through this quickly, but basically this is a type of data set that really focuses on different bacteria. So once we kind of had this bacterial culprit, we really wanted to focus in on that community. And essentially what we found is, so on the bottom axis there where it says pre-inoculation, those are all the samples that I talked about before we actually exposed anybody to anything. And so what we could see is that in those samples, there's very, very little, if any, Vibrio pectinicida. And then after we expose them in blue, those are all of our wasting sea star samples. So we see this big jump in this bacteria. And then again, along the bottom there in orange, those are all the healthy ones. So we see this massive difference again after exposure. This bacteria is very, very highly abundant in those sea stars.
Yeah, it's just so sad, once you finally kind of get it, know what it is, and you're like, now what, right?
Yeah, very satisfying in a lot of ways. Yeah. Yeah.
Yeah. I mean, that was, we now had a culprit, and we were thinking, gosh, what we need to do. One of the things that's sort of really useful about working with bacteria is that you can grow them outside of their host. And so, especially for sea stars, we don't have cell lines for sea stars. So if you wanted to grow virus, you basically have to grow it inside a sea star. There isn't another option, at least that I know of. And so since we had a bacterial pathogen, we then realized that we could grow this. And so we wrote to all of our diving friends, because we, at the time, when we figured this out, it was January, and it's not really the time of year that tends to be a lot of wasting disease in the field. Tends to be
older.
Yeah, exactly. And so we wrote to everyone, and we're like, if you see a wasting star, please let us know. And it turned out that there was sort of a disease outbreak that happened in one of our collaborators' labs. And luckily he told us about it. So this is Jason Hoden at Friday Harbor Labs. And, he was one of the people we'd written to. And so we jumped in a car and drove as quickly as possible, and arrived. These two sea stars are ones that he had in the lab. They were isolated. And they're at basically that early twisting stage. So you can see them, so their arms are all twisted together. And so we got samples from them, brought that back, and cultured Vibrio pectinicida from those samples, which is pretty incredible. Like, I mean, it's one of these moments, like, it was an entirely different time. It was a different lab. It was, we had used other samples and other things to find this culprit. Then we go down and get somebody else has a disease outbreak. We get some tissue from that animal, come back, culture what we find in there. Mm-hmm. And it's the thing that we found previously. It was one of those, I don't know, science doesn't normally work that way.
No, but it's just, super satisfying to see that even across these different areas that you're able to truly identify that this is the bug.
and to do through collaboration.
Yeah. I love
Yeah.
the citizen inclusion of sort of the citizen scientists or other, field scientists that were witnessing and could help in this effort.
It's been, yeah. This is like, I'm always trying to do collaborative research, and I would say the Sunflower Star Research Group that is currently, I think there's something like 60 to 90 of us.
70.
There's a bunch of us that are throughout the coast, and just incredibly collaborative. Like,
Great.
everybody is we're all yes people. So
Well,
Definitely a success story for collaboration. Like on this slide, there's like 3 different people that helped. Jason, who helped us get these samples from Friday Harbor, the culturing work was done by Amy Chan at UBC, the pathogen work there, that's sequencing to confirm that we have the right species in all of the cultures that are growing. That was done by Kevin Zhang at UBC. So like, yeah, just so many people coming in and
but having said
using
that,
their expertise.
put the pieces together. You can get lots of pieces of the puzzle gathered, but you actually did the synthesis and saw the patterns, and bravo.
Yeah.
thank you. Wow.
Team sport.
So then you had your pure culture there from the University of Washington, and you kind of did the repeat of what you had done before to verify.
Yeah, so this is like kind of the final step in like really conclusively demonstrating that one thing causes one disease. Because here, like all we're exposing them to is just this bacteria. So all the other cells and microbes that might be within the tissues that we had to use previously, those are all factors that we can eliminate when we're working with these cultures. And we reproduced the same patterns on the same trajectory. Like it was very consistent with what we've been seeing in the lab and in the field. So that was kind of closing the loop up in a very satisfying way.
But it looks like you had one that didn't die.
Yes, that happened. That did happen occasionally. Normally we had really high mortality in our sunflower stars. Occasionally we had a survivor in an experiment here and there. It gives me hope that there's resistance. Like that is a full question that needs to be evaluated and tested, and something that we're actively working on now.
Right. The natural resistance to the bacteria.
Yeah, it's very possible. And if it's out there, like we want to find it.
I don't doubt that you will. Tenacity and grit is
Oh yeah.
So these are the little
This is it. Yeah. This is like the culprit. Yeah.
So
yeah, sorry, go ahead.
I was going to ask if it was just, specific to kind of attacking or having a mortality in sea stars of various kinds, but here you see oyster scallops and others are also affected by it.
But not even in the same kingdom.
Yeah, it's not other echinoderms.
Yeah. We do like to be, I guess, careful. The Vibrio pectinacida was originally found in scallop larvae and an oyster larvae.
I'm sorry, not in the same phylum.
Yeah.
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 But they and then we found Vibrio pectinacida in sea stars. But Vibrios are they're capable of being the same species and having 2 strains not co-infect the same species. So we don't know yet whether the strain that comes from a scallop would infect a sea star, or whether the strain that comes from a sea star infects a scallop. Like I was like, I will say that we have a long list of what we want to do next.
About other echinoderms like urchin.
Sea cucumbers.
Yeah.
Yeah, exactly.
Yeah, I don't know.
My sense is that if they did if that was a big pathway, then we should have seen urchin and sea cucumber die-offs during the star die-off, and we didn't. I mean, we have seen sea cucumber die-offs, we have seen urchin die-offs, but not kind of at the same time or in the same scale as the sea star one. So but that doesn't, but we don't know. Is
the other
part.
alternative is like they could be reservoirs, right? Like there could be a lot of species out there that can harbor this pathogen and maintain it without actually dying from it. So, and those are, yeah, all questions that we need to answer to be able to manage what's going on as best as we can.
Status, what's happening?
Oh, there's still disease in the field. So at this point, like it depends on the species. So for pies aster, there's kind of seasonal disease outbreaks, I think basically everywhere. That tends to be in the warm seasons. So we tend to see seasonal outbreaks that are around August, September. But they're often only 5 to 10% of the population. Every couple of years, you might get a 20 to 40. And then we do have some evidence that pies aster is actually resistant. So ochre stars are actually resistant. Like we had in an experiment, one individual that we exposed, it got sick and then it got better.
Wow, that's impressive.
Yeah, and so we're very hopeful, and that pattern's happening. For pycnos, in BC, we are still seeing disease outbreaks. And like a year ago, we went to a site that had just like an incredible sea star diversity. We went originally in May, and then we went back there in October, and there was a massive disease outbreak. Wow. And lots of sea stars were dying. Came back the next year in May, and just like it was like horrendous. It was like pycnos were practically gone, but also all these other sea star species were gone. Like the diversity declined drastically. It's all sort of happening right now.
Yeah, that's frustrating.
Yeah, and like these are data from the field. Like I think these are from 2023, and this is wasting disease outbreaks in the fjords that I was talking about that are the refuge populations. So we're like, even in the fjords where there's a refuge from sort of the massive loss, we're still seeing wasting outbreaks.
Yeah.
Is there any correlation with the increasing acidification of the ocean that might favor the bacterium in ways that are subtle, but really enough to kick things over the edge? I mean, they might be there all the time, but when the chemistry of the ocean changes or the temperature changes, they're winners and losers. And some of these creatures that are there in small numbers proliferate. And they wouldn't if the chemistry of the ocean had not changed, but we're seeing a lot of things that are kind of not what they used to be.
Yeah.
It's a good question. I don't think we know at this point. So that's a solid question for us to look into. Add it to our list.
Especially when you see a bacterium begin to appear out of nowhere, probably was there, but with changing chemistry, their time has come, or temperature, or both. Maybe we have a bit of both, and it may not just be acidification. That's just one obvious factor that is being recognized globally, but there may be something regionally that we're putting into the ocean, or that's happening because of some complexities we haven't figured out yet.
Yeah, so these relationships I think are so dynamic and like so subtle sometimes, and like actually teasing apart what causes what, like, in these kind of trophic cascading.
But you're great getting to the bottom of this mystery.
Yeah.
I mean, it'll unlock our ability to do a lot of the interesting science that answers questions about acidification and temperature and all of those relationships now. So
yeah. Do you know how deep these guys go? I mean, I wonder if they're sort of in below diver depths. If maybe we need to get a submersible out there and look for them in deeper water too.
They do. I was like, I think they've been documented to 200 meters. There is, when you get to a certain depth, you start to be worried about rhizophalan versus Pycnopodia because they look somewhat similar. But we've done a little bit. we've done a little bit of ROV and we've done a little bit of drop cam looking deeper from the sites that we work on, especially in the fjords. Fjords are quite deep. And haven't seen a whole lot of evidence that there's at least high densities deeper, but they certainly are there. So
Yeah. I'm wondering if it's like, because obviously the deeper areas are colder. So kind of a retreat behavior to get to colder water to stave off the infection.
But might their prey be less abundant?
Yeah, for sure. They have a harder time finding something to eat.
Yeah, that's actually, I think like one of the ways I think about this, and I try not to anthropomorphize too much in how I'm describing it, but I think they don't know to avoid warm water. Yeah. Essentially. So it hasn't been around long enough as a disease. And part of the reason that I actually think the fjords are a refuge is that one of the sort of extra pieces of information is that fjords in the summer during the hot time periods have freshwater lenses on the surface.
Yeah, I was wondering about that too.
And so the freshwater lens keeps the sea stars out of the surface water because they don't like fresh water at all. And so like I watched them walking away from the water as the freshwater lens
really fast too. It's so funny to see those guys moving.
Exactly. So like see the freshwater lens coming and down they go. And so I actually think about them as trapped in the cold water when they're in the fjords, essentially trapped in an environment that is better for them. But if that wasn't there, they'd go up to the surface, probably because of the food. Yeah. And certainly they're not, when on the outer islands, if they were to just go down, the water would be cool, right? Yeah. They have access and like reasonable distance access to a better habitat that they wouldn't die from this disease. And so I think it just hasn't been around long enough for that behavior to have been worked into the system.
I think that's true for a lot of animals, birds, insects, whatever on the land, even fish, that the speed, the acceleration of change does not give enough time for kind of evolution to happen.
Yeah. So this is sort of just showing the places that we have seen Vibrio pectinacida in samples so far. So you can see in Washington, in British Columbia, and then I think, Alaska. And Alaska as well. And I like the sad truth of this is the reason we don't have stuff further south is because they all died. And we need to go back into the archives and get pre-mortality data.
Yeah. No, that's really tough because I remember kind of in that first map you had 100%, 99%. So it's really tough to find them.
Yeah, it happened so fast too. It was like one of the kind of cool things I think about this epidemic is like how fast people actually reacted and started to make something happen. Like this started in 2013 and like immediately people were like putting together emergency funding to try and get samples and monitor what was going on. And I think that's a really incredible thing that happened for this system that probably doesn't happen for many disease outbreaks and like was totally necessary because it just happened so fast and spread so rapidly. And so I'm actually quite impressed with the amount of data that was collected and the amount of observations that happened and how fast they were able to happen. And, I think it's something that we need to think about and work towards for, future epidemics or outbreaks. But still, like there's so many gaps that are missing because did we study sea stars intensely before they started dying in the billions? Like, no. So, no. There's a lot that we'll never be able to recover, but
right. But I think because they are such an iconic animal that everybody that goes to the shore or it's just kind of you think about ocean and people think about sea stars.
And sharks.
And sharks, yeah. Dolphin. But this is sort of the explosion of the urchins when they don't have their predators.
Exactly, yeah.
Well, there are other predators that we also prey upon.
Oh yeah, all the rockfish, sheep's head, wolffish,
otters,
kelp bass. Yeah, just they have a huge list of things that eat urchins, but we also eat a lot of those things
that would normally be along with the sea stars, keeping just the right number of urchins. We need the urchins. We need the whole system, but it needs to be in the proper, each one doing what they do
Yeah.
instead of us coming and destabilizing.
Further.
Yeah.
Yeah. Well, it's like you were talking about the die-off of the urchins in the Caribbean, and then all of a sudden all the reef would start to get overgrown with algae. But at the same time, they're also eating all the parrotfish,
Yeah.
which would normally come in and kind of
Enjoy the feast.
enjoy the feast, right?
Yeah.
So that because of these destabilization of the environment, we really have a lot of unintended consequence.
Wow.
Yep.
Oh.
Oh,
Yeah, this is sort of a reminder that there's so many other species, which is the sort of the other thing that we're working on right now is we've been collecting coalomic fluid in the wild from a bunch of other species of sea stars just to, you know, show that BPEC is also making them sick if it is, to evaluate that question. And we have found like early data, but we're finding BPEC in wasting other species.
Are there efforts to try to keep in cultivation some of these species that are fast disappearing? Well, we couldn't pose that question 50 years ago, but the cultivation of a lot of invertebrates now, including corals, people have mastered the fine art of figuring out what to do.
Yeah. So that is actually, so the collaborator Jason Hoden that we were talking about, he figured out how to raise Pycnopodia from like the sunflower stars. He can spawn them and then raise up adults from them. So grow the sunflower stars, which hadn't been done. So that happened after wasting happened. He figured out the system, and now there are lots of different places, different aquariums that are raising sunflower stars, and so we work with the Nature Conservancy on a lot of these projects, and they're really helping us coordinate around these ideas of raising animals. We're working to see if we can answer this question of if any of them are resistant. Any sunflower stars are resistant, because we might be able to raise resistant ones.
yeah, and then
in the 1990s, well, before then, the idea of raising jellyfish was
like
oh,
have you lost your mind?
You can't do that.
Everybody does it. Yeah.
Very, very far.
Somehow, just in case, worst case, that some of these creatures really disappear in nature.
Yeah.
There's at least the potential for getting them back someday.
Yeah. Oh yeah, we've seen stories with birds and other species where they could eventually
California
condors or scarlet macaws, things like that. But it to your point, it really all hinges on protecting and restoring the natural systems, healthy systems, so you can put these guys back.
Yeah, exactly.
All the pieces.
Yeah, all those computer pieces.
Yeah, exactly.
Yeah, exactly.
Keyword. Yeah.
Yeah.
No excess.
They've even, so like part of this group, this is actually part of the like Association of Zoos and Aquariums, like some of our collaborators that are part of that. They've actually figured out how to cryopreserve embryos and from sunflower stars. And so there are actual, backups and of cryopreserved.
Yeah, exactly. All right. Well, we've got a few questions stacking up here.
Great.
Susan said, I missed why healthy, abundant southern U.S. coasts are gone along the central and below. So I think what she's asking is, how is the distribution kind of stacking up of where populations are healthy or not?
Yeah, I mean, the pattern is that we saw the biggest losses in California and Mexico, and also Oregon, honestly, with upwards of 100% loss in that area. It could be temperature related. Seems probable that it's it's just a little bit warmer, and we do have we're working on evidence that this particular pathogen grows better in warmer temperatures, but doesn't necessarily have to be super warm temperatures. And so like warmer temperatures that are very much within the temperatures that they experience in their waters, at least currently.
Yeah.
And so that's likely part of what's driving that. I will say that for many years, there were essentially no one had seen a sunflower star in California. But in the last 2 years, they've seen like maybe 20, which is really hopeful. And also, you have to remember that there used to be billions.
Billions, yeah. Billions and billions. So
but it's not zero.
Like other stars.
So should we be encouraging sort of recreational divers to report sightings to you guys?
Yeah, and iNaturalist is a great place. Or and if you go to seastarwasting.org, they also have a good. It doesn't have to be a wasting star. You can also report, I think, just seeing a sea star.
Yeah, it seems like there's so few in some areas that seeing one is like a huge cause for hope, right?
Well, it's like
exactly.
You can get people to be starfish
watchers,
starfish, whatever.
Yeah,
watching whether they're healthy or not, just to know who's who and where they are.
Absolutely. Yes.
Helpful.
So Michael, it says, I might have missed this, but the causative pathogen is a bacterium, not another organism. Yes.
Yeah, it is a bacterium. Yep.
Lots of them.
And Keith says 5.75 billion is a huge number. We had maybe tens of thousands as a high estimate in Monterey. We would see a few on a dive. It seems they would likely be on top of one another. Where does the 5 billion or 6 billion estimate come from? Are there other areas where they are found deeper and come up to shallow water?
So the 5.75 billion estimate comes from, it was actually the Nature Conservancy got a group of people together that monitors sea stars from Mexico to Alaska. So the estimate is from Mexico to Alaska. And we basically looked at all of the data sets that we had and looked at it over the time frame from 2013 to I think it was 2017. And so it was like, given what we knew so we actually had data sets that went before 2013 because that was when the outbreak happened. And so given our estimates of populations before that time, how many did we lose? And that's how many.
Yeah.
Did the census of marine life have data that turned out to be helpful?
Oh, it's been just long enough since I looked at our data sets that I'm not sure if I know, but I mean, I know that we looked around at a lot of different data sets.
2010.
Yeah.
Anyway, just a thought. We need more people documenting what's out there.
Yeah. And having a place to put it.
Yeah.
Yeah.
Where we can share.
And access.
And compare. Chris is asking, how are sea stars impacted by bottom trawling?
Yeah. Well, I know they get picked up in bottom trawls. In fact, actually, one of the answers to your questions about our knowledge of the deep sea changes are that our bottom trawl data shows the decline in picknose as well. So like the data from scientists doing it. So I would assume if they're not being put back, that it's probably not helping. But I don't have any hard data on that.
And they're long-lived.
Yeah, you say decades old. So anyone lost in a trawl that's got a big impact. It's like an orange roughy, right? 100 years. So
who eats the starfish?
I mean, especially once they get to a half a meter to a meter wide, I'm not sure.
Like nobody messes with them.
We were just talking about that this afternoon because this year we have an individual. It's like probably this big, like maybe 3 years old, and it's got along 3/4 of it nice long arms proportional to its central part, and then the other quarter is about 8 or 9 little baby regrowing arms, as if something came and like took a bite out of it.
Taste.
So we were having that question of like, what could that have been? Like, what would eat these things?
Yeah, I think there's a lot of fish that they come and take a taste. So whether it's a wolf fish or a little shark or something, the cookie cutter shark,
Yeah.
So Keith has a question as well here. It says the urchin bloom happened in 2011 when sea stars died off in early 2014. Urchins were already 3 centimeters. Why is sea star wasting disease and die-off of sea star predators considered the most common narrative cause of purple urchin proliferation when we started noticing it 3 years prior? Um, it elevates the importance of sunflower stars to the point where people think kelp restoration can just be achieved by reintroducing sea stars. So,
What was the question?
I guess it's more of a comment. Yeah.
It's like a two fact. So sunflower stars definitely important. Sea otters are kind of the other half of that equation that eat a lot of urchins and definitely help control kelp forests, and also a species that has disappeared from certain areas of the coast. So I think it's kind of an impact of like a double whammy. And some recent research has shown that actually sea otters and sunflower stars, they don't necessarily select the same size of urchins. So you've got sea otters that tend to select the big ones because they're diving down and they're getting the largest ones. And sunflower stars, yeah, that might go for more like mid-sized or juvenile sunflower stars would eat the juvenile. So I think it's like definitely, and there's probably more at play there than even just those two, but there's certainly two like top predators. And in some areas like California, you see complete loss of both of them. And in other areas, you might have one or the other kind of thing.
Right. We also noticed that in areas where there's a lot of pressure on rockfish, that there's a lot more urchins because a big rockfish will eat urchins and small rockfish eat small urchins, even larval ones, or just the little landers. So just that kind of intense pressure, I think, on the so-called ground fish, and lobsters. Lobsters eat baby urchins too. So you can go back and look in who's it? Milton Love and his Love Lab. He has like all the documentation, stomach contents, and it's like urchin, 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 urchin in all these different fish stomachs. So I think it's like kind of that death of a thousand cuts, right? There's multiple impacts, and when things are out of balance, then it just goes wild.
Yeah, it's certainly not the only thing that has changed in that time frame.
Yeah,
but it's certainly one of the most obvious.
Yeah.
And one of the cool things about all these programs that are now captively rearing sunflower stars is like we're learning so much more about them as a species. So like we've had people saying, they can feed a juvenile sunflower star unlimited urchins and like they'll eat like 8 in a day. Like they'll just go for it, right? So like little bits of information like that I find so fascinating that like we're learning quite a bit, and they enjoy a diet of urchins.
Yeah.
So when they spawn, is it an annual thing or is it sporadic? What is it when these?
Yeah, I love to answer these questions because I'm always like, the answer is we didn't know. I mean, there were published dates for spawning for Pycnopodia, but I think it's actually less detailed than we needed to know. So
They spawn into the water.
They do spawn into the water. So I was like, a quick side note, I don't know what our timeline is. We're quite close. But a quick last minute side note, but it's also quite hopeful, is that this year I managed to be diving during a mass spawning event in a fjord.
Awesome.
Yeah, it was incredible. We went down and saw one spawning and was like, oh, that's amazing. And then saw another spawning. And then I think we saw 30 spawning Pycno. And that day, they're like big Pycno spawning. It was absolutely incredible. And that was in May. So they spawn in May sometimes in the British Columbia. But Jason sees them in Washington. He can have them spawning in like February, March. And then I think in Alaska, sometimes they spawn in June. So like I think there probably is some variability.
Yeah, it'd be great to kind of get all those data points kind of in one cohesive place where you can kind of look at some of the changes at the time and people could really collaborate.
We're working on that. That is yeah, exactly. I was like, we have a data set that we're putting together with a collaborator, Sarah Graveham. And actually, I think I can't remember, like we've only have like 60 observations of Pycno spawning in that data set. And that's, with us trying to get as many people as possible. So I guess if anybody's watching and has Pycno spawning information, do let us know.
Absolutely. And I think it also highlights the importance of some of these field laboratories, like, where both of you guys have been working there at the Hakai Institute. You're kind of like right on the water, can really access the regions and the Friday Harbor Lab. And so those field stations are so important for the active science and action to keep going on.
Yeah.
Yeah, exactly.
More causes for hope.
Good excuse to go diving.
That's right. That's right.
Exactly.
Well,
Get in the water.
We are up at the top of the hour. But thank you both so much.
This was really fun.
Yeah, absolutely.
Great. Thank you so much for inviting us.
Yeah, before we close, we'd like to say thank you to both Alyssa and Melanie and our producer, the Ocean Elders, and to everyone out there in the diving community who keeps showing up for these conversations. Water connects us all, and we're eternally grateful.
Yes.
So remain curious, spend some time out in nature, have a wonderful holiday season, and we're going to be back in January.
And remember,
Yes, remember, take care of the ocean.
as if your life depends on it, because
It does.
it does.
It does. And the sea stars need it too. Bring back that big guy again. He has to close.
The real star here.
The star of the show. There he is.
I can wave bye.
You guys. Bye for now.
Bye. Thank you.
Bye.